Hello, welcome to the CIRS Healing Collective expert guest video series. I'm Jenny Johnson, the host and facilitator of the CIRS Healing Collective, which has been a resource for aspiring CIRS overcomers to share and connect from around the world since 2020. I'm a shoemaker proficiency partner and CIRS coach, and most importantly, an overcomer myself. What you're about to see is a 10-minute excerpt from a 90-minute group discussion that members of the CIRS Healing Collective got to have live with this expert guest. If you would like to see the full 90-minute replay, come on over to the CIRS Healing Collective. We have a robust video library from the past 5 or 6 years of so many expert guests. Better yet, you'll be able to join our future monthly live discussions with experts to come. We'd love to have you.
But it also got me thinking, because we asked a few microbiologists, we understand when mold forms grow, they release compounds into the air. When we breathe them in, we get sick.
Yeah.
So we saw an association, though, between the presence of actinos in a living space and human illness. But what was the bridge? What's the link? What are actinos releasing? And microbiologists will tell you, we don't know. That they've never studied actinos through the lens of an environmental exposure. Mm. So I got really interested in beginning to do my own research on actinomycetes. A few things are interesting about them. Number one, they are classified as bacteria, but they have almost equal phenotypic or structural elements that look like mold. That they actually ride the line between a fungal form and a bacterial form. And they have sort of influences from both. The other piece is that there are 6,000 varieties of actinomycetes. They're very hardy, they're very robust, they're quite ubiquitous. They can live in dry environments, such as, almost desert-like conditions, as well as in completely watery environments, and everywhere in between. So one of the things you realize is that if you have a completely dry home that was originally built for you, doesn't mean that you can't get actinos growing. They can still be there. Harder for mold to grow, but certainly actinos can be present. So we began to understand the link between their presence and illness and Gini results. But what was the bridge? And I came across some soil science research that was very provocative, because what the soil scientists saw was that certain types of actinos, you can extract different kinds of enzymes and use those enzymes in industrial processing. I didn't realize that actinos were actually robustly used in all sorts of processing methods, including winemaking and all sorts of things. So what's used from actinos? There's an enzyme that they produce, and it's a subset of actinos. It's not all actinos, but many of them will. They produce an enzyme called beta-glucanase. And a beta-glucanase is a small enzyme that, when liberated or released, will bind to the outer cell wall of mold, and it releases beta-glucans. Why is that important? Because beta-glucans are probably the most inflammatory compound to our immune system that's ever been discovered. Wow. That when you measure macrophage response to beta-glucans, macrophages become so stimulated, they start behaving in ways that we never realized they could behave. So macrophages are part of what are called the innate immune system. And these are sort of blunt responses. It's thought that it's general, it's very inflammatory, but also there's no memory involved. Memory happens on the other side of the immune system. But if you stimulate a macrophage enough in its most extreme form, macrophages develop memory, and it's called trained immunity. And what happens is, once you train macrophages to have that memory, the next time you stimulate them with not just beta-glucans, but lipopolysaccharides, endotoxins, inflammagens, mycotoxins, you get a 6 to 7 times robust immune response. It's amplified. So once the macrophage is primed with beta-glucans, now when you encounter food compounds, VOCs, stress, inflammation from the gut, all sorts of other exposures, we get this enormous response. And this is what we see in our patients. The pathway that I'm describing is called the dectin pathway. And it's very unique and only recently discovered, the link between a beta-glucan stimulus and this trained macrophage immune response. And what's really interesting is when you look at the molecules that are stimulated along the dectin pathway, guess what? It's TGF beta 1, it's hypoxia-inducible factor, it's IL-6. It's all of these molecules that we see as part of that original work that Richie did. And now, so when I presented to Richie sort of what I think is going on, that actinomycetes are probably working in tandem with mold forms to release beta-glucans into the atmosphere. And this is likely one of the priming events. Mm-hmm. And now we stimulate trained immunity, and that's why people get so sick, because they're so inflamed. And we're seeing results of that, especially within Genie. We see, these gene sequences beginning to express related to the dectin pathway. And even Richie had this moment of, oh my gosh, I think you filled a big piece of the puzzle, which is why we're doing a research study now where we're measuring beta-glucans in the air. And we're trying to understand the relationship between a beta-glucan exposure and the CIRS model. One of the things that's interesting is we're seeing that there are a number of patients who might have clean surfaces. Their Hertz-Me score, their swab actino levels look okay, but they're still sick and they're not passing their visual test. Why? Because the beta-glucan levels are still high in the home. What that means is that you might do small particle cleaning, which is fine, but if the house is not breathing properly and the air is still contaminated, you're still going to be sick. Mm-hmm. And one of the big I think oversights that we've had is we've relied so much on swab sampling, but the truth is everybody who's sick from CIRS, it's because of what they breathe in. Mm-hmm. And we've stopped testing that. So we found a better test method where we can measure smaller molecules than normal. We can measure beta-glucans, which is why we're doing the new study. And I think this is going to provide a lot of insight on what's really going on in our living spaces. What are the additional compounds that are stimulating people to make them sick? What are the new pathways that I think are really being described as the most inflammatory, and why the body becomes so damaged? And because of the trained component, the memory component, no wonder it's so hard to break out of this cycle. Yeah. Because the body is so deeply primed to respond. And it's very hard to turn off those macrophages. So I'm helping Richie build a new way of looking at CIRS. And for one reason or another, I was sort of the first person to get there with him. So I think he kind of liked it that, he's getting help and getting new insight. So it takes a village, but this is one of, I think, the new insights that will grow in its importance. And there are others as well, but that's to me, a really novel one that I'm particularly interested in.
Yeah. I'm going to see if I can summarize that in really simple language and see if I got it. Because this is the first time that I've heard a little bit about these meters, these air quality tests, that are testing for beta-glucans, but I didn't appreciate the background that you just shared. I want to make sure I've got this, and maybe this will help others who aren't sure that they're grasping this too. What you're saying is that mold, one of the byproducts of mold is beta-glucans, and beta-glucans are very inflammatory. So we think of maybe mycotoxins a lot as a byproduct of mold, and let's test for mycotoxins, but you're saying beta-glucans are a highly inflammatory byproduct of mold, and they can be stimulated to be produced in the presence of actinos. So when there's mold in a home, maybe some mold spores and also actinos, the actinos prompt the release of these beta-glucans from the mold into the air. We may end up with a fine Hertz-Me or ERMI, because it's not necessarily, the structures in our dust, the mold structures or the DNA in our dust that we're picking up. But the beta-glucans, how can we assess for the release of beta-glucans? Again, I'm kind of liking it to mycotoxins. Mm-hmm. Mycotoxins in the air, but not mycotoxins, beta-glucans. So there's a way of that you guys are looking into developing of testing for beta-glucans in the air as another way of assessing a living environment, as opposed to just doing the dust sampling and, of course, other inspections and things like that.
That's right.
Wow,
So
wow,
yeah,
fantastic.
yeah. So I think, we're circling back. So I want to be able to measure the air mass in addition to surfaces, in addition to
Wow.
skin. And I think, through all of those methods, we're getting closer to understanding, at least the microbial component. But again, if you look at the dectin pathway, once it gets primed, there are other non-microbial compounds that will further stimulate it once those macrophages are turned on. And I think that's why you have these subsets of patients that can't stand chemicals or
Right.
odors or
Yeah.
foods